Chapter 6, Hemoglobin. Hemoglobin is a highly specialized intracellular protein within the red blood cell. Its function is to transport oxygen from the lungs to the tissues and facilitate the carbon dioxide transfer from the tissue to the lungs. It occupies 33% of the volume of the red cell, and it accounts for 90% of the red cell's dry weight. Each of the cell contains about 28 to 34 picograms of hemoglobin, and this is measured by the red blood cell index, MCH. Most of the hemoglobin synthesis occurs in the polychromatophilic normoblast. 75 to 80% of the hemoglobin is made before the red cell's nucleus is extruded. 20 to 25% is made by the residual RNA and mitochondria in the reticulocyte. Hemoglobin concentration is a result of a fine balance between red cell production and destruction. Here we have an image of the actual hemoglobin molecule, which is composed of 4 polypeptide subunits. Each subunit has a heme group and a globin chain. So heme is a tetrapyral ring with a ferrous, that means iron, in 2+, in the center of the molecule. Each heme subunit can carry 1 molecule of oxygen bound to the central ferrous iron. Each hemoglobin molecule can carry 4 molecules of oxygen. The globin chains include 2 identical alpha-like chains and 2 non-alpha-like chains, like beta, delta, or gamma. Each consists of a heme group surrounding the globin chain. They are all held together by salt bonds, hydrophobic contacts, hydrogen bonds, which form this tetrahedral formation. Let's look and dissect the hemoglobin molecule. On the left, you see that the quaternary structure of hemoglobin with 4 protein chains, and they're each folded around the heme molecule. They want to protect this molecule. So we have, again, 2 alpha and 2 non-alpha chains with the heme structure in the center. Now on the right is the heme molecule itself, which is the center of those globin chains, which is composed of a peripherin and an iron in the center. Now the iron has 6 coordinate sites. Nitrogen atoms of peripherin occupy 4 of those sites in a square arrangement around the iron. Iron is in the ferrous state, meaning 2+, and has 2 other coordinate sites, one of which is occupied by nitrogen on the proximal end, with another being attached to oxygen. Hemoglobin is synthesized in a complex series of steps. So looking at the mitochondria, the heme part is synthesized here and the cytosol of the red cells, while in the globin portion parts are synthesized by ribosomes in the cytoplasm of the cell. Production of hemoglobin continues throughout its early development from the pronormoblast to the reticulocyte in the bone marrow. At this point, the nucleus is lost, and even after the nucleus is lost, residual ribosomal RNA allows further synthesis of hemoglobin until the reticulocyte loses its RNA after entering the vasculature. Globin synthesis is directed by 2 genes on chromosome 11 and 16. Chromosome 16 zeta is synthesized very early in embryonic development, It is replaced by the alpha chain synthesis after about 8 to 12 weeks. The non-alpha globin genes are found on chromosome 11. These chains, the beta, gamma, and delta chains. The globin synthesis occurs in the polyribosomes in the cytoplasm of erythroblasts. Globin chains are released from the polyribosomes and are folded. They create a hydrophobic pocket near the exterior surface of the chain where heme is inserted into this pocket, and its job is to be readily accessible to oxygen. An alpha chain heme subunit and a non-alpha heme subunit combine to form a dimer. Two dimers combine to form a tetramatic hemoglobin molecule. Beta chains have the greatest affinity for the alpha globin chains, then it's gamma, then delta. Look at table 6.2, the normal types of hemoglobin according to the developmental stage of the patient. So first we're going to discuss hemoglobin F. This is a predominant hemoglobin during the liver and bone marrow erythropoiesis in the fetus. 90 to 95% of hemoglobin F is made until about 36 weeks gestation. Then after birth, it drops to 50 to 85%. Hemoglobin F is made up of 2 alpha chains and 2 gamma chains. Now in the adult, hemoglobin A is the major type of hemoglobin produced. It is composed of 2 alpha and 2 beta chains, and this is formed as early as 9 weeks gestation. The beta chain is produced at a lower level until the 3rd trimester of gestation. It does not exceed the alpha chain synthesis until birth. Beta chain production increases while the gamma chain production decreases. Hemoglobin F production is less than 2% of the total hemoglobin. It's very restricted to a few red cells, and these cells, called hemoglobin F cells, constitute about 2 to 5% of the adult red cells. Normal levels of hemoglobin A are over 95%, and they're reached after the age of 1 year old. Hemoglobin A2, which is 2 alpha and 2 delta chains, appear later in fetal life and take up less than 1% of the total hemoglobin at birth. In a normal adult, this hemoglobin A2 takes up 1.5 to about 3.7%. It has a slightly higher affinity for oxygen than hemoglobin A, but it has the same effect to release oxygen with using 2,3-BPG. Glycosylated hemoglobin, hemoglobin A1, is a minor component of A that can be post-translationally modified, which is known as fast hemoglobin or glycosylated hemoglobin. Here, the hemoglobin A1, either A, B, or C, especially hemoglobin A1C, is clinically most important because older red cells contain more hemoglobin A1C than younger ones due to exposure of glucose concentration. So the young red cells exposed to high levels of glucose for several hours can increase the hemoglobin A1C. Now this is a very good indicator as a control of glucose levels in diabetics because it's proportional to the average blood glucose level over 2 to 3 months. So you'll see in diabetics, you'll have an average hemoglobin A1C level of 7.5%, where a normal individual without a highly sustained glucose would be 3.5%. The function of hemoglobin is to transport and exchange gases. When blood reaches a tissue, oxygen diffuses out of the blood to the tissues, and carbon dioxide diffuses into the blood from the tissues. So oxyhemoglobin is a term to describe hemoglobin with bound oxygen. Deoxyhemoglobin is hemoglobin without oxygen. The amount of oxygen bound to hemoglobin and released to tissues depends on certain things like affinity for oxygen. The ease at which hemoglobin binds and releases is called the affinity of oxygen. So as the affinity for oxygen increases, hemoglobin has a higher ability to attach to oxygen and will bind. At this point, it does not release it easily. When there is a decreasing affinity for oxygen, usually at the tissues, hemoglobin or oxygen is released from the hemoglobin molecule. The term deoxyhemoglobin is describing when there is low affinity within the molecule to release oxygen. That is a tetramer in a stabilized subunit in salt bridges where it's in the tense state. Oxygen is not able to be picked up. It is released at this stage. When we talk about oxyhemoglobin, this is in the relaxed state. Those salt bridges are broken and the affinity to pick up oxygen is higher and it's able to conform and hang on to oxygen. Now we know oxygen binds to hemoglobin at a ratio of 4:1. One oxygen binds to each heme group of the tetramer, and depending on the interaction of the 4 heme groups, the T conformation or tense state in deoxyhemoglobin has a low affinity for oxygen. So loading the first and second oxygen molecules does not occur easily here. And as oxygen is released by hemoglobin to the tissues, the heme pockets narrow, they restrict entry of oxygen, and the space between the beta chains widen, where 2,3-BPG binds again in the central cavity. Here we see the molecule of hemoglobin where within the heme structure you see that 2,3-BPG binds in the central cavity of the deoxyhemoglobin or tense state. The cavity is lined with these positively charged groups on the beta chains that interact with the negative charges of 2,3-BPG. The alpha globin chains are in pink and the beta in blue, and the heme groups are in red, so you can see where the molecules are located. Discuss a little bit about carbon dioxide transport. So CO2 diffuses into the blood from the tissues once oxygen is delivered. So a small amount of carbon dioxide is dissolved in plasma and carried to the lungs. The diffused out carbon dioxide is expired or breathed out when it reaches the lungs. So here in the deoxyhemoglobin state where it's tense, it is able to bind carbon dioxide. It reacts with the uncharged N-terminal groups of the 4 globin chains to form carboxyhemoglobin. And in the lungs, the plasma CO2 is decreased, so the carbon dioxide bound to hemoglobin can be released at the lungs. It diffuses out of the red cells into the plasma and expired. Now, when we talk about aging red cells and the destruction of red cells, we normally think of extravascular hemolysis. Here, the red cells are removed from circulation by macrophages. The recycling portion occurs as such. The globin is broken down and recycled into the amino acid pool. And heme is cleaved by the macrophages as well. The porphyrin ring is cleaved, resulting in a release of the molecule of biliverdin and carbon monoxide. Carbon monoxide is released into the bloodstream, carried to the lungs, and breathed out. Biliverdin is reduced to bilirubin, which is released and bound to albumin in the plasma. Bilirubin is carried to the liver and conjugated, okay, called direct bilirubin, and here it is excreted in the bile. It's carried to the intestinal tract, metabolized to urobilinogen, and is excreted in the feces, or reabsorbed and later excreted in the urine. Now, when we talk about intravascular hemolysis, this is the breakdown of the red cell within the vasculature. Now here, because the hemoglobin molecules are intact, they have to be dissociated. So they have to be broken up from their alpha and beta dimers. So this quickly binds to a protein called haptoglobin in a 1-to-1 ratio. This complex is very large to be filtered by the kidney, so it's carried to the liver, where the hepatocytes take up this compound and process it similarly to how they would using extravascular hemolysis. Now we talked about normal hemoglobins. We're going to talk about acquired or non-functional hemoglobins. These are altered types of hemoglobin molecules which compromise the transport of oxygen. And this usually causes hypoxia, which is an inadequate amount of oxygen at the tissue level, and a physical cyanosis. So you see the bluish coloring of the skin. Acquired non-functional hemoglobin is called methemoglobin. It's very clinically important because it keeps iron in the ferric state, or the iron 3+ state, where it cannot bind to oxygen. It has a very increased affinity of remaining normal hemoglobin, which decreases the oxygen carrying capacity of the blood. So normally methemoglobin is formed at about 1% per day, but when you have certain conditions, this is greater. So let's talk about the inherited variant of methemoglobin. This occurs when there's an amino acid substitution in the globin chain near the heme pocket. Now here it stabilizes iron in the oxidized or ferric state, which cannot pick up oxygen readily. The congenital defects in reducing systems happens usually at birth, and the abnormality is in the NADH reductase pathway, which accounts for greater than 60% of this occurring. At birth, you can see that these babies are bluish in color or have cyanosis, and the levels can reach up to 20%. Now, the acquired types of methemoglobin include exposure to toxic substances that can oxidize large amounts of hemoglobin. And here you see this in about 30 to 40% of cases. Sulfhemoglobin is a stable compound where the red cells carry it until the cell is removed from circulation, but a sulfur atom binds to the periphery of the porphyrin ring of the heme group, which results in a drastically right-shifted oxygenation dissociation curve, which leads to an ineffective carrying of oxygen. Sulfhemoglobin can combine with carbon dioxide to form carboxy-sulfhemoglobin. Normally, levels don't exceed 2%, but cyanosis can be seen with even 4% of sulfhemoglobin. It's associated with occupational exposure to sulfur compounds, environmental exposure to polluted air, or to certain types of drugs. Carboxyhemoglobin forms when the molecule is exposed to large amounts of carbon monoxide, which is incapable of carrying or transporting oxygen. Hemoglobin has an affinity for carbon monoxide 200 times higher than it does for oxygen. So here you're seeing carbon monoxide occupying the space where oxygen should be. This is seen in increased levels in smokers. Acute carboxyhemoglobinemia can cause irreversible tissue damage and death. Now 6.5 is a good chart to show you the abnormal acquired hemoglobins, what their acquired change is, and how we can detect it.